Hello, everybody. Good afternoon. Welcome to webinar 5 of Platomics IVD Ready program, entitled GSPRs are challenging, standards are beautiful. The aim of this webinar is to provide orientation for laboratories in understanding the general safety and performance requirements of the in vitro diagnostic regulation. Platomics is a company based in Vienna. Our mission is to bring safe and effective innovations to market quickly and efficiently. We digitize regulatory processes for different stakeholders in vitro diagnostics. Today, we have also our new release of the GSPR Assistant. The new feature creates the GSPR checklist very quickly and also takes components of the workflow into account that are integrated via the Workflow Studio. But more on this exciting release a little bit later. My name is Simone Latkolik. I'm leading the customer success team for labs here at Platomics. Today we have invited following guest speaker and expert. I'm very happy to announce Professor Kurt Satlokal from the Diagnostic and Research Center for Molecular Biomedicine at the Medical University in Graz in Styria in Austria. Professor Satlokal has in addition led several European Union projects, for instance, the FP7 funded project SPEDIA, focused on developing European standards and norms for pre-analytical processing of tissue samples for molecular testing. As an expert on standards, he will talk about the standardization of pre-analytics and sample quality. Andreas Oberleitner, he's head of quality management and regulatory affairs at Platomics, has experience in this field for different IVD manufacturers for more than 20 years now. He's especially an expert in the domain of conformity assessment for medical devices. Andreas will give an introduction to the Annex 1 of Article 5.5 of the IVDR, the GSPRs, and will later give a demonstration of our new release. You can always ask questions in the QA section during the webinar in an anonymous way, and we will try to answer them by the end of the webinar, if we have enough time. For health institutions, as you might know, the IVDR came into force on May 26 in 2022 with the first set of requirements for in-house developed diagnostic tests. Diagnostic laboratories are to have fulfilled the Annex 1 of the IVDR, which is the GSPRs. The next set of requirements will become effective in May 2024, and here labs have a few new GSPR-related tasks. My colleague Andreas now will explain this in his presentation.
Thank you very much, Simone. Good afternoon from my side. I would like to tell you a little bit more and give you an introduction into the topic of general safety and performance requirements. For that purpose, I prepared a few slides and I would like to share my screen. Just give me a second, please. I would like to talk a little bit about regulatory landscape. We had these slides already in the past webinars as well, but for everyone who is new to our sessions, I would spend some words on that topic again. So first of all, we know that there had been a strong transition from the old IVD directive to the new regulation that was already 2017. In the past, laboratories and health institutions in general had been excluded by the old IVDD explicitly. So therefore, they have been seen only as so-called operators of devices, but not as manufacturers. Now it's completely different with the IVD regulation. The regulation explicitly mentions laboratories, in that case, health institutions in general, as being not only operators, but also potentially manufacturers if they are creating in-house devices. In-house devices are regulated via the Article 5.5 within the IVDR and come with specific requirements on it. We also know that there have been in the past also publications which changed a little bit the timeline. It's worth mentioning that they never changed the requirements themselves up to now. They only changed the timeline that led to a certain rollout transition period. We will talk about that a little bit later. We also know that as being a health institution or laboratory, we need to consider national laws like the medical device laws, for example, which might be peculiar to each and every member state within the European Union. And we also need to consider guidance documents, especially one had been published in the beginning of the year 2023, the MDCG document, which should be quite well known in the meantime. But that's not yet all. We also need to consider applicable standards. Why? Because it is always expected to work according to the state of the art. And standards like guidelines or also common specifications play a major role in defining the state of the art. So state of the art is always changing over years. That also means that standards are changing. And it's always the obligation of a stakeholder to identify potentially applicable standards and probably make use of them. One of these standards, I think, is from specific interest. This is a standard which is still under draft status, but hopefully or potentially will be published quite soon. This is an ISO standard, which is focusing fully on the so-called LDT, the laboratory developed test concept, and comes with common requirements. We also know that there could be many scenarios leading to an in-house scenario. So it's not only the case where you really develop your own device, literally, which would be the scenario 5 in that graphic, but it could also be the case that you probably repurpose devices which are originally intended only for research use only. With that step, you're entering the realm of in-house IVDs, and that means that you take over the responsibility for that devices, and you need to fulfill the relevant requirements and do the relevant documentation, of course. We also know that the MDCG document gave us more clarity on what is not deemed such a device, and therefore also, of course, is not deemed as being an in-house device. We know that, for example, protocols, written procedures can be exchanged without a problem. Those are not deemed an in-house device, while the devices themselves used with this protocol, of course, still might be in-house devices. The patient specimens are not in-house devices, and also results in the form of reports, for example, can also be transferred between health institutions because they are not seen as in-house devices, just to round it up and make it clear. I already mentioned the timelines. So we have 3 major milestones in gray, blue, and black here. The gray ones are already in force since May 2022. We know that we soon will see a next wave of upcoming requirements, all the blue ones, mainly dealing on the one hand with quality management aspects and requirements, on the other hand with requirements for additional documentation, also publications and stuff like this. And last but not least, in the year 2028, there will be a last requirement which requires laboratories or health institutions to justify why they are still using in-house devices in the case if there might be similar or equivalent CE mark devices already on the market. Maybe you have also recognized that in the past week there has been a proposal for an amendment. So potentially this timeline, the last one, this milestone, could potentially be extended for 2.5 more years, but this still needs to be decided over the next weeks. Today, we will focus on the first point because this is already the main requirement. In-house IVDs must comply with the general safety and performance requirements, abbreviated as GSPRs, which you can find in the Annex 1 of the IVDR. So one more time, I would like to remind you on the Article 5. It says, with the exception of the relevant general safety and performance requirements set out in Annex 1, typically in-house devices or laboratories are excluded from the rest of the IVDR. So that sounds pretty easy, but at the end, is it really the case? And it's immediately clear that, of course, this is not that easy, because this Annex 1 mentioned here is already literally the heart of the whole regulation, and it describes all requirements which are needed to be fulfilled if they are applicable for your devices. So that means a lot of investigation, potentially also a lot of data creation, and of course, a lot of documentation. Another issue here might be that typically, in many cases, these GSPRs are formulated in a very vague and general way. Why? Because these requirements, they need to be suitable for 25,000 different types of products. So of course, they can only be on a very high level and can only require very basic things without telling you how to fulfill this. To give you just an example, I have one GSPR 17.4, which requires you to design your product in such a way to provide a certain level of intrinsic immunity on electromagnetic interference, such that an adequate level is achieved and to let the device operate as intended. So you see already there's a lot of wording in it, which is more or less a little bit blurry and grayish. Yeah, and the answer on that, of course, is there might be potentially standards helping you in fulfilling the details and giving you hints how to fulfill these things. Additionally, we also know that Article 5.5 comes with several more requirements, as we have seen in the timeline list already. So what is the dilemma now? From our many discussions with laboratories, we have seen that there are many questions arising around the GSPRs. For example, what are these GSPRs, or where do I find them? I think we already answered parts of that. What exactly is my device under assessment? This still is sometimes a bit problematic to identify. In laboratories, we are more working with workflows than with individual devices. How can we assess them? So what need we do here? That has to be done in a structured and also systematic way, of course. What can I do to fulfill them? We already mentioned standards, for example. And how should I document this? So to sum this up again, what are GSPRs? We know that GSPRs are the general safety and performance requirements. They are written down in the Annex 1 of the in vitro diagnostic regulation. They are applicable not only to manufacturers, but also to health institutions right now with their concerning their IHIVDs. We know that the Annex 1 consists of 20 main requirements, but if you break it down into the sub-requirements, then you quickly find out that there are more than 190 detailed requirements you all need to have to check, and you have to analyze and assess, and then also to fulfill if applicable. We also know that the national competent authorities are obliged to assess the GSPR fulfillment from the laboratories. So that might be different in different countries. Therefore, in most cases, it's the same national competent authority, which is also responsible in overseeing the general medical device market, but that might be different in each case, and you need to check it with your own national competent authority. Last but not least, to remember you one more time, this already needs to be in place since May 26, 2022. Yeah, how does it work? Just a quick overview. We know that our regulatory framework consists of many things, but mainly the IVDR. Let's focus on the in vitro diagnostic regulation right now. And one part of the IVDR is the GSPR. On the other hand, we have the health institution or laboratory. They typically also have implemented a quality management system, in many cases already accredited as well. And if you have an in-house device, you need to assess each and every of these general safety performance requirements. For that purpose, you typically set up a document, which is typically called a GSPR checklist. And this GSPR checklist needs to contain all the GSPRs and your results and assessments to it. So on the other hand, that means that you probably have already some verification documents which could match to some of the GSPRs, or in cases of gaps, you need to add potentially additional verification documents. In many cases, you probably even have to create new data when it comes to performance evaluation, for example. So doing that gap analysis leads to potentially to some gaps or open points here. And then the question is, okay, how can I fulfill them? And here again, we definitely think that it is worth digging a little bit into the standards topic or guidelines topics, because how to fulfill the GSPRs, that can be in many cases really facilitated by applying suitable standards. So to sum it up, GSPRs more or less define what to fulfill. The standards, guidelines, and so on help you how to fulfill these requirements. And last but not least, your evidence documents, which, by the way, play a major role or an unseparatable part of your technical documentation for the in-house devices, represent the proof how to fulfill these requirements. So very quick summary on the structure, not to dive too much into details. The GSPRs consist of 3 main chapters, as you can see here, with different topics: general requirements, more device-related and safety-related requirements, and last but not least, in chapter 3, you find requirements on labeling, on information, and instructions for use, etc. So very quickly, you find some requirements on what to document exactly. There you can borrow the requirements from the Annex 2, because they describe pretty well what is expected to be found in such a documentation. On the one hand, the explanation why a requirement is applicable, and especially why it is not it is not applicable if the requirement is not applicable to your device. Which methods, for example, which harmonized standards you're using to fulfill a requirement, and last but not least, to have a precise identity of the document of the evidence, and also a link to the location, if suitable. So very often we see the approach, which might be very typical, to write up a long document, for example, a Word file or an Excel spreadsheet for that. Please keep in mind that with that, you still need to talk about 190 requirements and fill each and every of these empty cells. Well, what is coming next? A very quick glance on the May 26th deadline I already mentioned, 2024. One of the requirements dealing with GSPRs are, for example, that we also need to write up now a declaration. So a laboratory now needs to write a declaration. Please keep in mind that this declaration, of course, is signed with a certain power of attorney. Someone who is signing the document really confirms to the public that these GSPRs are fulfilled, and is also more or less responsible for the completeness, correctness, and also for all the given justifications. So I think it's really worth to have a good GSPR checklist and a good derivation of justifications, arguments in the background. And last but not least, please do not forget that this declaration has to be made public so that everyone can find it. Another thing I would like to mention here is that competent authorities can request detailed information how to fulfill the GSPRs on request. And the MDCG document again gives us some insight what might be expected. For example, data on the design, on safety and performance, and on expected benefit from the device, among some others. Good. And with that, I would like to stop my theoretical part and would like to hand over back to Simone.
Thank you, Andreas, for this really nice introduction to the topic. Now, I would like to hand over to our expert talk. Professor Kurt Satlokal will talk about standards and GSPRs from a lab perspective. He will talk about the standardization of pre-analytics and sample quality. We have pre-recorded his contribution to the webinar and are showing a video now due to appointment conflicts on short notice.
Yeah, ladies and gentlemen, it's my pleasure to talk today about the role of standardization of sample pre-analytics and sample quality in context of the development of in vitro diagnostic medical devices. Before I go into details, a brief disclaimer. I have R&D collaboration with QIAGEN and invited as lecturer by Roche and AstraZeneca, and I'm also CEO of a spin-off of our university, Zatlocal Innovations. Just to introduce the importance of the topic. Please be aware that diagnostic errors are still a major challenge in patient care, and still diagnostic areas contribute to a significant number of patient deaths and adverse events. What is often not known that about half of these diagnostic errors are based on errors in the pre-analytical phase. So the role of the pre-analytical phase is very often underestimated. And this gained also now new relevance in context of the in vitro diagnostic regulation, which is in force since May 27, and now there is some extension in the implementation based on the risk of the device. But very important is that it has to be shown what is the scientific evidence, what's the analytical performance and the clinical performance. And now in the IVDR, there is explicit reference made in context of the analytical performance that all information on pre-analytical parameters have to be shown and have to be part of the technical documentation. And here I show you the some of the specific references that are made in IVDR on this. For instance, it has to be shown in context of performance testing. The specimen type shall describe specimen types that can be analyzed, including the stability, such as storage, where applicable specimen transport conditions, with a view on time-critical analysis methods, information on the time frame between taking a specimen and its analysis, and storage condition as duration, temperature limits, freeze-thaw cycles, and so on. This information can only be generated if samples with a defined quality were used in generating performance data. Because if this information is not known and this is not collected and documented, you can never describe under which pre-analytical conditions the performance of your IVD is guaranteed. There is also reference made to these aspects also in the Annex 1. The general safety and performance requirements again highlighted the specific reference that the analytical performance has to include the determination of appropriate criteria for specimen collection, handling, and control of known relevant endogenous and exogenous interference substances. Important to know that this has also to be addressed in context of so-called laboratory developed tests. Another reference on the Annex 2 on the technical documentation. Also here you have to show what are the appropriate specimen types with information including on the stability, storage, specimen transport conditions, time critical analysis, and so on. So you see IVDR makes several references to pre-analytical parameters. Now the big question is how to demonstrate here this information, how to provide this information. Because IVDR as a law never goes into these details, but here we can get guidance on different elements. There could be harmonized standards that were explicitly harmonized for the applicability for IVDR. There could be common specifications. There could be guidance documents for the medical device coordination group, but also other SEN and ISO standards are relevant because they define state of the art and therefore have to be considered also in the CE conformity assessment process. And so I want to share with you a little bit more details on what standards can be used with regards to determining pre-analytical parameters for IVDs. Please be aware when we talk about pre-analytics, this is the complete process from collecting a sample from a patient to the transport, the stabilization, the further processing, the isolation of analytes, analysis to making a diagnosis. And the pre-analytic part includes all the steps except the analysis and making the diagnosis. And there are a series of standards that are relevant that cover these aspects. For instance, the overarching quality management standard 15189 for diagnostic laboratories, but there are also a series of specific standards for the preanalytical phase that all give reference to the general quality management standard 15189. And these are families of standards like the 21661, 1, 2, 3, or the 21664, which describe the preanalytical steps for different sample types and different analytes. And as I said, these are families, so they are very similar in the structure and content because the goal was to have common workflows and only make deviation where it's essential to address quality requirements for a specific sample type or an analyte. And here's the examples of these standards. I don't want to go into detail. It's just for your information and documentation. You see here for the different sample types like frozen tissue, FFPE tissue, blood, but also body fluids, saliva, circulating tumor cells. They are all addressed by specific standards for the various analytes. Now, I want to introduce to you these standards on one example in more detail. That's the standard for analysis of isolated DNA from formalin-fixed paraffin-embedded tissue, which is, by the way, the most commonly used biological sample for molecular testing. Here, these standards distinguish two phases of the preanalytical process: the part that is outside of the diagnostic laboratory and the part that is inside of the diagnostic laboratory. Why is this important? In general, most laboratories have well-established and documented workflows in the laboratory. They have laboratory information system, but many organizations are lacking proper documentation and standardizations of the steps outside of the diagnostic laboratory. So what happens with the sample from the patient, e.g. in the operations theater, until it's transported and it reaches the certified laboratory? And these standards cover both elements, which is some challenge in the implementation because it really requires collaboration of different institutions or organizations like diagnostic laboratories and surgical departments. That's the table of content you find, and what you already can see here, that these standards describe in much more detail elements and steps in a diagnostic workflow than a law like the IVDR. And they give general background information like the scope, the normative references. In this case, they refer to the 15189, provide terms and definitions and general considerations. And then they go into detail and describe all the steps outside of the laboratory and then inside the laboratory. And they describe what information should be collected on specimen collection, the transport, processing, and so on, and then the various steps inside the laboratory. I cannot go into the detail, but just want to raise awareness what information, what level of detail of information you may get in these standards. Very important being aware on the principle of the standards. These standards are no SOPs, so they are not specific for a laboratory, but provide general guidance how you have to standardize a process, but they don't describe exactly what you have to do, because the principle is you have to show whatever you do is fit for the purpose, for the intended use, and this has to be shown. So they don't describe that you have to use a certain collection tube or a certain isolation kit. Whatever you use, it has to be according to the intended use. and this has to be described by the manufacturer or shown by the diagnostic laboratory in case it's a so-called lab-developed test. It requests that procedures are standardized. You have to document the compliance with the procedure, and there are only very few examples where the standards make a specific reference to a reagent or a method, and I will show you this in a minute. For instance, the standards define the effect already of warm ischemia and cold ischemia, which poses some challenges in the implementation, because warm ischemia during surgery already starts within the body when, for instance, the blood supply of an organ is disconnected in tumor surgery, and very often this information is not documented, or at least not accessible to a diagnostic laboratory. And so these new workflows request that this information is documented and is available and is also shown in context of the parameters under which the performance of the assay is guaranteed. After warm ischemia is the cold ischemia. That's the time when a biosample is removed from the warm environment of a body until it's stabilized. This could be either by freezing or fixation. Also, this has to be documented because major changes may occur during these both periods. And there is much scientific data that demonstrate and show the impact on an annulid in these two phases. And that's also reflected in the standards. They give reference and raise awareness that the molecules may change. So it is not only degradation of biomolecules, because in general, cells and organs are still viable when they're removed from the body, so they respond to this challenge and ischemia condition. So very often you see a very active stress response due to sample processing, which is not related to the original disease condition. Another very important element is the effect of formaldehyde fixation. Formaldehyde modifies proteins and nucleic acids, but also very important that formaldehyde gets oxidized to formic acid. And this results in a change in the pH, which enhances modification of nucleic acid. And as a consequence, you may see major impact on formaldehyde fixation, and there is a major need for standardization of formaldehyde and the fixation procedure. And this is just one of a few examples to show you how marked these effects are. In this case, tumor samples from breast cancer were analyzed after frozen, immediate freezing after removal of the body, or after fixation in formaldehyde. And here was the expression of different tumor-related genes analyzed. Each dot reflects a different transcript. The analysis was done by RT-PCR, and the yellow triangles are the CT values for the various genes obtained from the fresh frozen sample. The blue squares are the corresponding CT values from the RNA isolated from the matched paraffin-embedded sample, the same tumor, the same patient. And you see here a major shift in the CT value of all, essentially all transcripts, although the same amount of RNA was used for reverse transcription. That's not a big surprise because the RNA is chemically modified, so you have a lower reverse transcription efficacy. But what was a surprise? That this effect is different for the different transcripts. So the concept that referencing a transcript to a housekeeping or control gene allows to correct for this variable is wrong, because your gene of interest may behave differently than a reference gene. Interestingly, we repeated the analysis 6 months later, and we found a further shift of this curve. That's the green signals. And this was repeated 3 times, complete independent RNA extractions, reverse transcription, PCR amplification. And you see that the analysis itself is highly reproducible. So obviously there is an aging process going on in the sample, showing that even a formalin fixed and paraffin embedded sample is not stable. It changes. And that's the problem when you use archived material for showing, or let's say the relevance of a diagnostic marker or for validation, that these aging effects have never been considered in studies or publications. And as a consequence, also the standards give reference to the fixation procedure. They describe a concrete recipe for formaldehyde and also give some warning on that water content may impact the stability of your sample, which could have major implications on a diagnostic assay. I'm running out of time, therefore I want to stop at this stage, just raising awareness on the importance, acknowledge the team that is working on these fields. What I showed was the result of many studies and projects we have done, and here I just acknowledge the project that provided funding and contributed to the knowledge of pre-analytical factors. Thank you so much for your attention.
Thank you to Professor Satlokal for this very interesting example and contribution to our webinar. Now I will share my screen. So with this, I'm very happy now to announce our new release of the GSPR Assistant. The IVD Assistant guides you through the IVDR requirements of your in-house developed tests step by step, starting with the intended purpose and then the documentation of each step in the, of your in-house device in the Workflow Studio. The Workflow Studio contains the product database where components of each workflow step can be integrated. Furthermore, it contains a database of standards. But this will be shown now by Andreas, who will give a demonstration for us.
Yes, welcome back. Thank you very much, Simone. Second time again, I would like to share my screen. Before entering the demonstration, I would like to summarize a few more things on our platform. As Simone already indicated, what do we want to achieve with the platform is to have really a one-stop shop for the entire regulatory process from A to Z. So in the future, this platform really should help you in doing all of your required regulatory affairs related tasks and steps and creating documents. The creation of a compliant documentation is, of course, the key aspect. We provide a variety of templates and information and so on. The Workflow Studio has been mentioned and also already been shown in the past, and I will also spend a few words later in the demonstration on it. The knowledge base to help you in performing your tasks at the point of care, so supporting you with additional texts, examples, and so on within the tool itself. We help you with providing wizards. Wizards are automatic programs with some kind of smart algorithms or rule-based algorithms in the background, helping you even in creating the generation of the whole content of the documents. It is a software as a service. You do not need to install anything on your local machine. Of course, we are fully following the relevant safety and security standards, cybersecurity standards. The data center itself is certified according to the relevant ISO standard as well. We do regular penetration testing, etc. Last but not least, talking about languages, currently the system is available in English and also in German, but additional languages are already planned and in development. Yeah, and maybe the best at the end, this version is also free of charge for laboratories and health institutions. Okay, this picture we have seen already. I just want to mention one thing especially, which is this product database. So what we also achieved in the meantime is to build up a product database in collaboration with many manufacturers. So that means that on our platform, you can already find a lot of products, and manufacturers provided their information which is relevant for the GSPR and also regulatory topics into this database. That means that every information that is already there, you can easily reuse, or let's say the system will reuse it and populate it automatically into your documents, which really saves you a lot of time in populating your own documents because the information can be processed by the system. So where do we stand right now? Everyone who had followed our past webinars knows exactly that we already provided wizards and templates for the intended purpose case. We already did successfully the qualification as an in vitro diagnostic and also documented it by the helping hand of the IVD assistant. We also did the risk classification in the past. And finally, we already were able to specify our workflow by defining the steps and also assigning these components and products. And as I already mentioned, the beautiful thing here is that these components do not come only as an entity into your system. They really come together with a handful of information provided by the manufacturers of these components. So therefore, you not need to, again, research and ask for all of this information and then putting that manually in a very cumbersome way into your documents. That all will be done by the platform automatically. And by today, we release a new module, which we call the GSPR Assistant. This is a module which will help you in work on your GSPRs for your in-house device. On the one hand, helping you in finding out about the applicability and also the justification to give you some suggestions on that. And last but not least, also in helping you defining standards which you would like to use to prove that you're fulfilling these requirements. And with that, I would like to say that we have spent enough words and directly jump into the tool itself. So what you can see here is directly the tool. So I am here in a demo environment. So please consider that all the devices, all the workflows you're seeing here are of course only for demonstrational purposes, so not real products for the sake of the demonstration. What we already did is providing information on general information on the workflow. We already went through the whole intended purpose definition. We already created documents for it and so on. We already passed successfully the risk classification. And for the use case I want to show you today, I would like to start again with the Workflow Studio. So maybe you remember from the last session, the Workflow Studio is a tool built into the IVD Assistant, and it helps you in defining the workflow you want to see as your device under assessment. So let's pretend we have a whole NGS demo workflow here. This workflow consists of 6 steps here. As you can see, we already pre-populated all of the steps with some demo products here. One product is still missing, so the system indicates that this last step does not yet bear any components or products. What I would do right now is I simply add a component by pressing the button. And what you see then on the right side, this is our whole database on products or components already available on the platform. Please again keep in mind what you see here are of course not real products. Those are only demo products here in that case. So we already prepared another demo product which is called Gene Analytics Sequence Harbor Pro. And I will now show you what you can do here. First of all, you can investigate that product. You see this pseudo product here is a software product because I would like to use it in my bioinformatics analysis step. It is a research use only clearance, so not an IVD at all. That's what I need to consider as well. The information is given in English, and the system also provides you with the information. And what you see here is typically already the information provided by the manufacturer for the product. So you have a good access to this important piece of information. Furthermore, there's a document shelf. And for example, the manufacturers can also provide you with very useful documentation. Of course, it's an RUO, so therefore is no declaration of conformity available, but there is potentially an instruction for use made by the manufacturer and maybe even more other documents like technical notes or whatever. Yeah, you would also find the contact information here if you have any questions to the manufacturer itself. But let's pretend that we want to use this component right now. I select this one. And if I scroll back down, then you see, cool, now this component is part of my workflow. So by adding this, we already know that in the background, a lot of steps are happening. So all of the information is now going to be processed. I believe that I'm now ready and feel like that I can complete the workflow, which I will do right now. And yeah, in the next step, if you're interested, you could already check the document which has been fully automatically created based on the given information. This is more or less a very general document. We call it workflow specification, but it collates all the information from the purpose, from the product description in general. So this is your own workflow product description, but it also collects all the information from the different steps, for example. So every step has a name and a general description. So this is more or less representing the SOP, if you'd like. And for each of these steps, you will find the basic information on the relevant components. So you also receive a nice bill of materials at the same time. Yeah, if you want to, you can also export this document. Just will do it very quickly just to test if it works. Yes, we have it here. And yeah, what you can do is with this PDF, of course, use it within your quality management system, sign it off, document it, and archive it. But back to the application. Maybe more interesting is that we have then another step which helps you also in analyzing if you're using these components according to the intended purpose and intended use. This is very, very relevant because changing the intended purpose and changing the intended use both might be indicators that you're using devices not according to the given prescription of the manufacturers, and therefore you're probably using it in an in-house scenario. It looks like follows. So this is already pre-filled because I did it already for the rest of the components. But in the end, you see that my new product, the Geno Analytics Sequence Harbor Pro software product, still needs some attention by me. That means that I need to check and also confirm, am I using this component according to its intended purpose? By the way, you can hover over these blue things and then you always will find a kind of glossary or explanation and helping text, what that means. Or am I using it not according to the intended use? So the intended purpose is really for what the device is intended to be used, given by the manufacturer. And intended use is how it is going to be used and embedded and combined and maintained, etc., etc. So let's pretend in our example that I'm not using it according to the intended purpose, which is clear because the device itself is an REO and I'm using it in diagnostic purposes. So therefore, the purpose is already changed by me as laboratory. But for this example, I would say I'm at least using it according to the intended use, so how I should use it. Still, I need to define more in detail why I'm not using it according to the intended purpose. I think that's very important to document. I'm not changing the testing population here. I'm not changing the intended specimen types, which probably are given within the manufacturer's IFU. But I will quickly note down, just as an example here, usage for diagnostic purpose. Just to document that I'm using it for a diagnostic purpose instead of using it for REO, save it and create it. And if I scroll down, then I see, then I see that it's now successfully completed. I can save it and close it. And with that, I gave all my statements, how to purpose, repurpose, or reuse, so to say, the original usage of the devices. And that can be defined in a grade of detail as you like. some of the wizards. There are even more wizards helping you in some specific cases, but for the sake of time, I will not go too much into detail here. The second step then is that I now can check my general safety and performance status. And entering the GSPR wizard, you will find that given by our algorithms in the background, miraculously, many things are already pre-selected here, and that's really the cool thing. First of all, let's pay attention for the yellow field here. In our example, one of the manufacturers did not provide GSPR-related information. Therefore, we always need to check these pre-filled fields already at selections with care. But at the end, many of the other manufacturers already did. And what you can see here is more or less an accumulated combination or pre-processed, pre-filled answer of all of these questions. So yes, it is for a medical purpose. We know that already from our original purpose. And we see that we have many yeses here because we also have many products here. So the chance to have applicability for many of these questions typically is yes. So what we need to do then is just to check that thoroughly and see if we agree to all of these answers. In our case, we would say yes. We say we are happy with those answers, save and create. And with that, actually, it's already done. So what originally would be a 190 count of questions would now be only covered by, let's say, a couple of questions. And if I now enter the next stage, which is the review stage, then I see, you maybe recognize, we have chapter 1 to 3, the requirements from the Annex 1. We see that in chapter 2, for example, we have several of the requirements already pre-filled as being applicable, some are not applicable, and so on and so forth. So the system already did the full applicability questions for you for all of the 190 requirements. But not only that, if I, for example, enter this example here, you would even find a justification text. So the system is so nice and provides you already with a justification for the applicability. It would also give you a suitable justification text for non-applicability if that would be the case. You can edit it if you want to, but for now I'm happy with it. Then you maybe see there are still open exclamation marks here, which warn you need to fill out or should fill out something here. For this example, 9.1a, we can probably say, I would like to add some standards. You can always add individual standards, but of course, in that case, I would like to show you our nice database on standards. You see there is a standard database at the moment consisting of approximately 200 standards where you can add standards if you believe you want to use them to prove this specific requirement 9.1.a. And I would say, for example, 15.1.9 could be a good match here because it definitely covers some points of validation in general. So let's add it here. And maybe even the second one, which we heard before. Professor Zatlukal mentioned the ISO 20166-3. Probably it could be a good idea to also follow this standard. And in our hypothetical example, we say, okay, we also followed this standard for the pre-examination processes for my DNA. And therefore, I will also add it. Okay, last but not least, we can also add evidence documents here. In our example, this is not yet part, but it's already a sneak preview. We have one document like this one, which is a validation report. This is just an example here, but I will reference to it. And here I can easily reference to it. I still need to do it manually, but as soon as this will be part of our product, of course, you can easily automate this as well. Let's say this is 2024-1-30, and we are in a version number, let's say 2.1. And now we have also added this document here. Okay, and then you see this question mark or exclamation mark disappears because we have filled all the relevant information. Yeah, and with that, this will help you in the next step. For the sake of time, I will abbreviate a little bit here to create your GSPR checklist. This will follow very, very soon in our tool as well. Just again, a sneak preview. How would it look like? So in that case, it would then look like this, that this is already from our templates we provided in the past, that the application text is in there, the standards I mentioned are in there, the evidence documents are referenced in there, and this document can also be populated completely automated. Good. Yeah, next steps will follow. Very soon we will come up with the next features, but for the GSPR wizard, to sum it up, we have now created a highly automated tool that helps you breezing through the 190 requirements from the IVD Annex 1, and hopefully also help you a lot because plenty of this information is already given and pre-filled, thankfully by the help of our manufacturer partners who provided this information. Yeah, and with that, I would like to stop the presentation and hand over back to Simone. Thank you.
Thank you, Andreas, for this really nice demonstration of our new GSPR assistant. So it took a little bit longer. I still have some information for you that I would like to share. Yes, if you haven't registered for the IVD assistant yet and would like to get access, you can contact us via email, or you can also register directly on our website. You will receive your user credentials within 5 business days from us. Also, if you have further questions or feedback, you can always contact us via email to info@protomics.com. During the IVD Ready program, we have already covered a lot of topics. In the next webinar, we will talk about validation, a big topic for diagnostic laboratories, and Protomics will release here templates for the validation in the IVD Assistant. The next webinar will be in March, entitled An Ounce on Validation. Also, we have to announce a new service that we are developing currently, the NGS validation services for labs that do genetic testing. Your lab may have done validation before. The new thing is now that your lab must document a compliant validation process, and to be ready to do so, to be ready also to provide it to your national competent authority if necessary. The NGS service helps you to set up the validation plan with all necessary parameters to execute your experiment and also upload the data. The result will be integrated in your PLATO-X IVD Assistant documentation. The NGS validation service will help you to set up your validation plan and report in 3 steps. Here we have a QR code, so if you're interested in the specific service, you can use the QR code on this slide. Or you can also contact us using the email address info@protomics.com. Protomics is currently developing this tool, and therefore we initiated an early adopter program for this service. Labs that are potentially interested in using this program starting already in March can register for the unique early adopter program using the registration link, or simply let us know via email. So we received a lot of questions in the QA section. Unfortunately, we are over time already. So if you ask a question anonymously, you can also write an email and to us, and we'll answer your questions. With this, I want to express my thanks to all participants. The participants who have joined us today and who have joined our program, the IVD Ready program by 2024. Labs can still join us, so you can also invite your colleagues and share the program if you like. A big thank to Professor Kurt Satloukal for his great contribution to this webinar, and Andreas for his demonstration, his nice introduction, and the demonstration to our new release of the GSPR Assistant. Thank you very much. And last but not least, a big thank to our team, especially to our product team, the software developers, and the regulatory team at Platomics. They do an amazing job. So without you, so thank you so much for making this program even possible. With this, I wish you a wonderful afternoon on behalf of the Platomics team.